friends, this is Preeti and you're watching my solution. In this video, I'll be teaching about the culture media. Culture media is important question for 10 mark question and also for 5 mark question. It has been asked in PYQ. In culture media, the important questions will be, like sub questions will be definition and classification and with the examples. What is culture media? Culture media is, it may be solid, liquid or semi-solid that contains the nutrients which are required for the growth and survival of the microorganisms. For the growth and the survival of the microorganisms, we need few nutrients that are present in this culture media. What all constitutes the culture media is, it contains water, electrolytes, peptone. Peptone is a mixture of partially digested proteins after partially digestion it forms peptones. And it contains agar, meat extract and yeast extract. Agar is a solidifying agent in the culture media. It is prepared by the seaweeds. Commercially, they are occurred as a powder form. Agar, for a solid media, we need agar 1 to 2%. For a semi-solid, 0.5% and 6% for proteus swarming. In addition to this agar, we add blood, that is sheep's blood, 5% sheep's blood or serum of the sheep. For extra nutrition, we add for fastigious bacteria, we need extra nutrition. We add 5% sheep's blood or serum. The types of culture media, there are two ways we classify culture media based on the consistency, that means solid, semi-solid or liquid broth, or based on the method of growth detection, conventional growth detection or automated growth detection. Conventional means we use manually we see the visualize and then detect the growth. It includes the various types of conventional culture media are basal media, enriched media, enrichment broth, differential media, transport media, selective media, anaerobic media. All media only comes. You need to remember the other names like enrichment, so selective, transport medium and differential medium, anaerobic. Conventionally means we just look at the bacterial growth and then we predict we see the growth of the microbes. Automated culture media means we for the blood and all we need sterility. There we use automated machines. The growth is detected automatically by this equipment. This is automated culture media. And now we come for the simple basal media. By using this simple basal media, we create other media like enriched media and selective media, transport media and all. Simple basal media, the minimum requirements for this is for the growth of the non-fastigious organism, non-fastigious bacteria, that is peptone. Peptone, peptone water. Peptones are the partially digested proteins. Peptone, 1% of peptone, NaCl 0.5% and water. This is a peptone water. For water, we add NaCl 0.5% and then peptone 1%. This is a peptone water. And nutrition growth. Growth for the peptone water, you add meat extract 1% to give extra nutrition for this peptone water. Nutrition agar, you add agar as a solidifying agent, 2% solidifier. Semi-solid medium is we reduce this concentration of agar to the 0.2 to 0.5%. Here 2% was there, you reduce it to 0.2 to 0.5%. This is all the simple basal media includes, that is peptone water, nutrition broth, then nutrition agar and semi-solid media. Semi-solid media is nothing but we are reducing the concentration of the agar, that's all. Like what we read, the definition of the culture media, that is solid, liquid or semi-solid which contain nutrients which are required for the growth and survival of microorganism, and it constitutes the water, electrolytes, peptone, meat extract, yeast extract and agar. Agar is obtained from the seaweeds and then the commercially available as a powder. It is acts as a solidifying agent. For a solid medium, we require more concentration. So 1 to 2% agar is required. For a semi-solid, 0.5%, and for a protein swarming, 6% is needed. Sheep's blood we acts as an extra nutrient. That is 5% of sheep's blood or serum. Types of culture media based on the consistency: solid, liquid, semi-solid. Then based on the method of growth detection, we have automated method, that is systems only, equipments only detect the growth of the bacteria, that is automated media. There is no manual thing is required, so they maintain sterility. The machines they do cleanly. And conventional culture media, they manually we detect the growth of the bacteria. Example: basal media, transport media, enrichment media, selective media, all these are examples for conventional growth media. Simple basal media includes a minimal the nutrients which are required for the growth of the non-fastigious bacteria. There's peptone water. Peptone water, to water you add 0.5% NaCl and 1% peptone. Then for nutrition growth you add, for the same peptone water you add nutrition content, that is meat extract 1%, and nutrition agar you add agar 2% agar. And then semi-solid you have to minimum the concentration of the agar, that is 0.2 to 0.5%. The uses of the basal media, what is the requirement? We are preparing basal media, why it is required? The basal media is used for growth of the non-fastigious organisms which requires a minimum nutrients. This basal media acts as a the base for the preparation of the other media. So we can by adding other things, we can convert that into other media. Nutrition growth used to study the bacteria. Nutrition growth is used to study the bacterial growth curve. Bacterial growth curve as already discussed. There I said about the nutrition growth. And nutrient agar is required for the oxidase test, catalase test, and the slide agglutination test. And to study the colony morphology, the pigment for the pigment demonstration also we require nutrition agar. This is all about the culture media. We studied about the definition and the constituents what are contains in the culture media, types of culture media based on two ways, based on the consistency and based on the culture media, how we detect the growth of the bacteria. And simple basal media, uses of basal media. This is all about the culture media which you need to write for a 5-mark question. Thank you.